Now we have a FOXG1 research update with Hourinaz Behesti, our Director of Science, and Nasha, our CEO. So tell us, let's start with gene therapy. That's what everybody wants to know. Where are we today with FOXG1 gene therapy?
So we have some really exciting data. We actually just came back from a conference, American Society for Cell and Gene Therapy, where we saw sort of have a broad overview of what's happening in the field. And for FOXG1 in particular, we have really exciting data coming out of a couple of labs that we have funded. So there's a study that has been done by the Lee Lab in collaboration with Nationwide. And I can't really go into the details yet. The data is preliminary, but it's looking really great, and so we're excited about it.
Yeah.
And we are talking about next steps, what we need to put in place to kind of move it forward as fast as we can. We also have another lab who are actively working on FOXG1 gene therapy at UT Southwestern, so we're also excited about that. And we saw at this conference actually that these labs that we are working with are really at the forefront of driving gene therapy for central nervous system.
Great.
So
yeah, exactly. I mean, both, Catherine Meyer gave a number of talks. Sarah Sinnett's lab, discoveries that have come out of that lab were talked about. And what's exciting is that these gene therapies that are being used for other disorders are in clinical trials right now, and we are seeing really solid data that is showing an improvement in some pretty measurable outcomes for patients. So we're in the same labs, we're learning from the same things that they're going through, and as Hourinaz mentioned, in our preclinical work that we're doing on animal models, we're seeing kind of similar progress. So it's giving us a lot of hope.
Great. And what about the screen projects that we have?
Yeah, so we have 3 projects on the way where we basically screen small molecules, and we're looking at different things, all sort of from rescue of what we call phenotypes in cells, and also in sort of like an organism, which is a zebrafish that we're funding. And so these projects are on the way, and they're each going to be screening a couple of thousand of compounds. So we're excited for that too, and we're hoping to have data starting to come out as early as this summer.
Great. And on the ASO front, this is something a lot of people want to know about. ASOs are very promising right now, exciting right now.
They are, and they are much cheaper to manufacture than gene therapies. They're also not a one-time treatment, so you have to go continuously, which is, a pain that our children will have to go through these different, to the hospital to get the medication every 3-4 months. But it's also really great because we can adjust for dosage and different things, and we're seeing a lot of promise. So Hourinaz, maybe you want to talk about what we have cooking with
so on that front, we have a new partnership with Crayon Bio, which is a biotech company who are basically interested in collaboration with the Lee Lab in developing ASOs for FOXG1. So we're really excited that they have taken on this project. It's a biotech company that is going to be really helpful in pushing it forward, basically. And then we're also about to actually fund a new project in Italy with Dr. Malamacchi, who's also identified some really promising ASO sequences. And he's tested them in human neurons, actually. And so we're excited to fund that project. He has some really exciting ideas about how to modify them to increase their, for them to have better delivery and also reduce their toxicity. So we're excited to push that forward too on that front.
Yeah, and this is an exciting project because we've been funding Dr. Malamacchi for a few years now, and the beauty of these scientists is that they've been working on the basic biology of FOXG1, really understanding the molecular structure, the pathways, etc. So these are the best scientists who tend to end up figuring out the right therapies because they are so ingrained in the actual gene and the disease that they start to notice that, hey, if we modify this pathway or this is something interesting we're noticing, we might see a therapeutic result. So that's why we've started really early with these scientists funding basic science, and now we're funding them to do translational projects where they're actually finding therapies. And it's because they understand FOXG1 so well.
Yeah,
so he's really an expert. I mean, he's been working on FOXG1 for 20 years.
Yeah, 20 years.
Wow.
And another lab that actually we are going to be funding is in Japan, which is another FOXG1 lab.
Yeah.
And he's more looking at sort of like circuits, and he's trying to Koichi Miyoshi.
Yeah.
So he's addressing a whole other set of questions, but has also like deep expertise in FOXG1. He's been doing this for 20 years. So
absolutely.
Yeah. And I'd say the other exciting update we have is Dr. Jean Paz at Gladstone Institute. So I don't know if you want to give a little insight there.
Yeah, so she's an expert on recording sort of like inside of the brain in animal models, EEG of like exactly how neurons fire. And so she's done some really excellent work in the past, and she has never worked on FOXG1, but we're excited to partner with her. And
it's about the genesis of the seizure, right? So that's what our parents want to know about. She's going to identify where the seizure starts.
Exactly. So the aim of the project is to look at different brain structures that we know are affected by FOXG1 syndrome, and trying to kind of hone in on it and identify and get a better readout of what is really happening inside of the brain when they have seizures.
Great. Okay, well, that's a fantastic update, and we'll keep them coming. And thanks for your support.
Yeah, thank you, everyone.
Thank you.